Good morning, students. Our today's topic is Megasporogenesis, that is ovule. Let us familiarize ourselves with the structure of a typical angiosperm ovule. The ovule is a small structure attached to the placenta by means of a stalk called funicle. The body of the ovule fuses with funicle in the region called hilum. Thus, hilum represents the junction between ovule and funicle. Each ovule has one or two protective envelopes called integuments. Integuments encircle the ovule except at the tip where a small opening called micropyle is organized. Opposite the micropylar end is the chalaza representing the basal part of the ovule. ओव्यूल की स्ट्रक्चर में ओव्यूल का जो स्टॉक है, जो डंडी है, उसको बोलते हैं फ्यूनिकल। और हर ओव्यूल प्लेसेंटा के साथ में अटैच्ड है। फ्यूनिकल और ओव्यूल के बीच में जो पार्ट है उसे बोल रहे हैं हाइलम। ओव्यूल के आसपास कुछ प्रोटेक्टिव लेयर्स हैं, उनको कहते हैं इंटेगुमेंट्स। ओव्यूल के पास कुछ कहीं जगह पर इंटेगुमेंट प्रेजेंट नहीं है, उस स्मॉल ओपनिंग को बोलते हैं माइक्रोपाइल। माइक्रोपाइल के ऑपोजिट पार्ट में चलाजा है। जो इंटेगुमेंट्स हैं उनके अंदर कुछ सेल्स हैं जिन्हें बोल रहे हैं न्यूसेलस। सो इनक्लोज्ड विद इन द इंटेगुमेंट इज अ मास ऑफ सेल्स कॉल्ड न्यूसेलस. Cells of the nucellus have abundant reserve food materials. Located in the nucellus is embryo sac or female gametophyte. An ovule generally has a single embryo sac formed from a megaspore through reduction division. Reduction means by meiosis. Watch the diagram, figure 2.7. They have in the first A diagram a dissected flower of hibiscus showing pistil. Stigma, style, ovary, thalamus is shown here in this diagram. B diagram, it shows a multicarpellary syncarpous pistil of papaver. Papaver plant की जो ovary है, many ovaries हैं, that is why multicarpellary. Many ovaries are present, that is why the term multicarpellary. Syncarpous means all the ovaries are fused. So which type of ovary or pistil is present in papaver? Answer is multicarpellary syncarpous pistil is present. Important. From diagram question can be asked. Second part, in C diagram, which type of ovary or pistil is present in case of mycelia? Answer is multicarpellary apocarpus. Apocarpus gynoecium. Gynoecium, many ovaries are present. Apocarpus means they are not fused. Multicarpellary, that is many ovaries are present. Apocarpus means these ovaries are not fused with each other. So in papaver ovaries are fused, and in mycelia the ovaries are not fused, they are free. D diagram is of a typical anatropous ovule. So first is on the top hilum. Hilum is that point which joins the funicle with the ovule. Then is funicle is the stalk. Stalk of the ovule is known as funicle. Micropyle is the opening where integuments are not present. In the ovule, two integuments are present, outer integument and inner integument. Inside the integument, nucellus, these cells are present. Few cells are present which provide nourishment to the embryo. Then embryo sac where egg is present. Opposite the micropyle is chalazal end, that is the basal part. So next is megasporogenesis. The process of formation of megaspores from the megaspore mother cell is called megasporogenesis. अगर एक मेगास्पोर मदर सेल से मेगास्पोर बनने के प्रोसेस को बोल रहे हैं मेगास्पोरोजेनेसिस. Ovules generally differentiate a single megaspore mother cell in a micropylar region of the nucellus. न्यूसेलस के माइक्रोपाइलर रीजन की तरफ जो कुछ सेल हैं वो अपने आप को मेगास्पोर मदर सेल की तरह वो फॉर्म कर लेते हैं, डिफ्रेंशिएट करते हैं अपने आप को माइक्रोपाइल की तरफ कुछ न्यूसेलर सेल्स हैं जो माइक्रोपाइल की तरफ हैं, उनमें से कुछ सेल अपने आप को मेगास्पोर मदर सेल की तरफ से डिफ्रेंशिएट कर लेते हैं। ठीक है, यूजली वन सेल होता है, वो सेल आपस में डिवाइड करेगा और वहाँ पे उस सेल में मियोसिस होगी। रिडक्शनल डिवीजन मीन्स मियोसिस। तो पहले वो 2 सेल्स फॉर्म करेगा और फिर वो 4 सेल्स और मेगास्पोर टेट्राड बना लेगा। अगेन इन द फिगर 2.8 डायग्राम इज शोन, पहले एक लार्ज मेगास्पोर मदर सेल बना। ये मेगास्पोर मदर सेल कहाँ से बना? न्यूसेलस माइक्रोपाइलर एंड की तरफ न्यूसेलस का कोई सेल अपने आप को डिफ्रेंशिएट कर ले रहा है मेगास्पोर मदर सेल में। उसमें मियोसिस हुई, पहले इस डिवीजन में मेगास्पोर डायाड बना और सेकेंड डिवीजन के बाद मेगास्पोर टेट्राड बना। टेट्राड मीन्स 4 सेल्स बने। इन मेगास्पोर टेट्राड में से 3 सेल्स डीजनरेट कर लेंगे अपने आप को, सिर्फ एक ही सेल फंक्शनल रह जाएगा एज शोन इन द डायग्राम। देन बी पार्ट, फर्स्ट इन दैट जो फंक्शनल मेगास्पोर सेल होगा उसमें न्यूक्लियस डिवाइड करेगा बाय माइटोसिस और 2 न्यूक्लियाई फॉर्म होंगे। फिर वो 2 न्यूक्लियाई अगेन माइटोसिस करेंगे, डिवीजन, और 4 सेल्स फॉर्म होंगे। फिर अगेन माइटोसिस के बाद 8 सेल स्ट्रक्चर फॉर्म होगी। सो अब एक सेल के अंदर 8 न्यूक्लियाई आर प्रेजेंट। इनमें से 3 न्यूक्लियस एक पोल की तरफ चले जाएंगे, 3 न्यूक्लियस दूसरे पोल की तरफ, एंड इन द सेंटर 2 न्यूक्लियाई विल बी देयर दैट फॉर्म द पोलर न्यूक्लियाई। Clear? 3 nuclei towards one pole, that towards the micropilar end, and 3 nuclei will move towards the chalazal end. Then wall formation will occur. So now 3 cells are towards the micropilar end, 3 towards the chalazal end. Where the micropilar end is present, that side one egg is there. And with the egg 2 synergid cells are there. Synergid cells, they have filiform apparatus. Some structures are present, they are known as filiform apparatus that guide the pollen tube towards the egg. Filiform apparatus का काम है वो pollen tube को egg की तरफ guide कर रही है. Inside, in middle of the cell is polar nuclei. The 2 nuclei which are left, they form the polar nuclei. On the chalazal end 3 cells are present, they form the antipodal cells. तो हमने क्या स्टडी किया? जो फंक्शनल मेगास्पोर बना था उसमें माइटोसिस डिवीजन होगी, 8 न्यूक्लियाई बनेंगे, 3 माइक्रोपाइलर एंड की तरफ जाएंगे, 3 चलाजल एंड की तरफ, और 2 बीच में पोलर न्यूक्लियाई बनेंगे। जो माइक्रोपाइलर एंड की तरफ होगा वो उसमें से एक एग होगा, और जो चलाजल एंड की तरफ होंगे वो 3 एंटीपोडल सेल्स फॉर्म करेंगे। इन न्यूक्लियाई के आसपास बाद में सेल वॉल फॉर्मेशन हो जाएगी। It is a large cell containing dense cytoplasm and a prominent nucleus. The megaspore mother cell undergoes meiotic division. What is the importance of megaspore mother cell undergoing meiosis? Meiosis results in the production of 4 megaspores. That we have studied that megaspore tetrad is formed. That is, by meiosis 4 cells are formed. Now the pleiady of this megaspore is haploid. Because reductional division has occurred, so the pleiady of this megaspore is haploid. Next is female gametophyte. In majority of flowering plants, one of the megaspore is functional, while the 3 other degenerates. Only the functional megaspore develops into the female gametophyte or embryo sac. The method of embryo sac formation from a single megaspore is termed monosporic development. What will be the pleiady of the cells of the nucellus? Megaspore mother cell, the functional megaspore, and the female gametophyte. Pleiady means number of chromosomes present. Nucellus will be 2n. Megaspore mother cell 2n. Functional megaspore n. And female gametophyte also n. Haploid number. Let us study formation of embryo sacs in a little more detail. The nucellus of the functional megaspore divide mitotically to form 2 nuclei which move to the opposite poles, forming the 2 nucleate embryo sac. Two more sequential mitotic divisions results in the formation of 4 nucleate and later 8 nucleate stage of the embryo sac. जैसा कि अभी मैंने बताया कि पहले उस फंक्शनल मेगास्पोर में माइटोसिस होगी और 8 न्यूक्लियस बन जाएंगे. It is of interest to note that these mitotic divisions are strictly free nuclear, that is nuclear divisions are not followed by immediate cell wall formation. अभी उनमें cell wall formation नहीं हुई. After the 8 nucleate stage, cell walls are laid down, leading to the organization of the typical female gametophyte or the embryo sac. Observe the distribution of cells inside the embryo sac. 6 of the 8 nuclei are surrounded by cell wall and organized into cells. The remaining 2 nuclei, called polar nuclei, are situated below the egg apparatus in a large central cell. So 3 nuclei towards the one pole, other 3 towards the other pole, and 2 nuclei are present in the center in the central cell. There is a characteristic distribution of the cells within the embryo sac. 3 cells are grouped together at the micropylar end and constitute the egg apparatus. All the cells which are towards the micropylar end, they are known as egg apparatus. The egg apparatus in turn consists of 2 synergid and 1 egg cell. The synergid have a special cellular thickenings at the micropylar tip called filiform apparatus, which play an important role in guiding the pollen tube into the synergid. So here question can be asked, what is the function of filiform apparatus? The filiform apparatus guide the pollen tube towards the egg or guide the pollen tube into the synergid. Synergids are the 2 cells around the egg. 3 cells are at the chalazal end are called antipodals. The large central cell, as mentioned earlier, has 2 polar nuclei. Thus, a typical angiosperm embryo sac at maturity have 8 nucleate and 7 cells. Can you see in the embryo sac? This is in the diagram 2.8c. That is the structure of a mature embryo sac. Here 7 cells are there. 3 cells towards the chalazal end and 3 towards the micropylar end, 6 plus middle cell. That is 7 cells are present. But how many nucleus? 8 nucleus are present. 3 towards the chalazal end, 3 towards the micropylar end, and 2 in the middle. So embryo sac in angiosperm has 7 cell but 8 nucleus. The middle 2 ones are the fused together. They are known as polar nuclei. I hope this is clear. Thank you.